سلام جميعا، أهلا وسهلا فيكم بميتينج ريسيرش بكونفرنس ريسيرش في باوندريز. قبل ما نبلش حكون عم نعرفكم هيك تعريف صغير عن الكلاب. أنا اسمي آية بشر، فايس بريزيدنت لنادي بايوس أمضة. ومين بايوس أمضة؟ هو نادي البيولوجي بكلية العلوم. تأسس سنة 2019، والهدف الأساسي نكون عم نساعد الطلاب ونعرفهم أكتر عن الاختصاص عن طريق الأورينتيشنز السنوية اللي بنكون عم نعملها، كمان نضوي أكتر على المجالات المتاحة لهذا الاختصاص. كمان من أهداف النادي نكون عم نعمل أنشطة ضمن كليتنا أو بالتعاون مع كليات تانية مثل مؤتمرات وورش بتساعد الطلاب بسوق العمل، طرق كتابة السي لينكد ان، موتيفيشن لتر، وكمان ايضا المشاركه بالمهرجان العلمي الكبير اللي عن جد عن طريق مختلف سكشنز قدرنا نفرجي مختلف مجالات البيولوجي. واخيرا اكيد ما تنسوا تتابعونا على صفحاتنا انستغرام، فيسبوك، لينكد ان، واتساب، حتى توصلكم كل الانشطه والفرص المتوفره لكم. بالنسبه لايفنت اليوم اللي رح يمتد لحد الأحد بكرة، أوكي، رح نتناول فيه مختلف العناوين مع سبيكرز طلاب جامعة لبنانية قدروا يتخطوا كل التحديات ويعملوا publications ويكملوا PhD. رح نبلش لليوم مع أول سبيكر، مصطفى خليل، PhD student in molecular biology بجامعة مونتريال كندا. رح يكون عم يحكينا أكتر عن التوبيك investigating the interactomes of the oncogenic fusions of sarcomas to reveal novel therapeutic targets. إذا أنت على السمع مصطفى خليل فيك تكون عم تبلش، تفضل.
Okay. Thank you, Aya, for the introduction. But as you know, guys, I did my master's in cancerology at the Lebanese University. Then I worked almost 2 years in the PCR COVID-19 diagnosis. اشتغلنا مع عليم بالجامعة اللبنانية واشتغلنا بمستشفيات تاني. Then I got the opportunity to pursue a PhD in molecular biology. It's a molecular biology program that is related to the Faculty of Medicine of the UDM. So I do my PhD at the research center of the hospital Maisonneuve Rosemont at the Cellular Signaling and Cancer Research Unit in El Bashir Affar Laboratory. So in my topic or in my thesis, by the way, it's my second year in PhD, and just to let you know, if you want to come to Canada, a PhD, it's almost 4 to 5 years. So in my thesis, I will be identifying the interactomes of sarcoma oncogenic fusions to reveal novel therapeutic targets. So maybe you are not very familiar with sarcomas. Sarcomas are the cancers of mesenchymal tissues. Mainly, they are the soft tissues and bone tissues. This type of cancer is like not common and it's not very frequent. It's kind of rare in adult among adult cancers, but it's very aggressive and it's very heterogeneous. Mostly we have the Ewing sarcoma subtype and the liposarcoma, which is like the cancers of the adipose tissues. Sarcomas can be divided into sarcomas with simple genomic profile and complex genomic profile. By simple genomic profile, we mean that it doesn't have a lot of mutations within the genome. So 30% of sarcomas are due to gene fusions, and 10%, in this 10%, we can find activating or inactivating mutations like P53 inactivation, or we can have MDM2. I don't know if you are familiar with these genes. And the remaining 60% are due to a very complex genomic alterations where there is no specific mutational patterns. However, the molecular mechanism of sarcoma genesis are not fully understood, so it's very interesting cancer, and it's worth it to go in depth to analyze, especially these fusions, because they represent 30% of these cancers. The survival rates of sarcomas are variable and depends on the tumor stage, and it can be, or the survival rate can goes up to 85% if the tumor is local or at stage 1, while the survival rate can decrease down to less than 10% if the tumor is metastatic or at stage 4. Uh, for example, based on the Canadian Cancer Society, they have their statistics, and sarcomas, they represent sadly 10 to 15% of pediatric cancers, and they represent only like 1% of adult cancer. But the question is, what is the significance of gene fusions on the survival rates of sarcoma? So I did some analysis, in silico analysis of some previous data for this subtype, which is called rhabdomyosarcoma, and I did the analysis for the survival rates to compare when we have a fusion versus no fusion. And you can see, for example, when we have a fusion, for example, we have PAX3 FOXO1 and we have PAX7 FOXO1. These fusions are very common for this subtype of sarcoma, and you can see clearly that the survival rates when we have the fusion are much lower than that when we don't have a fusion. Because so far, for these fusions, we don't have like specific drugs or therapies that can target specifically these chimeric proteins. So they are kind of difficult to target. So it makes sense why we have this lower survival rates. So by this, our aim is to go more and investigate uh, and characterize these chimeric proteins or these fusions to find some, I don't know if we can find like therapies, but at least to understand the molecular mechanism behind the pathogenesis and associated to these gene fusions. Moreover, the sarcoma mortality rates are increasing, and the annual percentage changes are increasing. And here I'm reporting the data of the Canadian Cancer Society. You can see that the mortality rates among hit top 10, top 2 among men, and it's the top 3 among women. And this shows that this type of cancer or is like emerging as, I don't know, a medical challenge, if we can say, because so far all the treatments that are used so far, they consist only on radiotherapy, some conventional chemotherapy, and there is like no specific targeted therapies against sarcomas. At the molecular level, two theories have been proposed to describe sarcomagenesis. Well, in the first theory, as you can see, we have multiple oncogenic hits that occur at the mesenchymal stem cell state, and then these complex genetic alteration will promote the development of one of these subtypes of sarcomas. In the second theory, we have an oncogenic hit that occurs at the progenitor cell state, and then by simple genetic alteration, we will have like the development of a particular subtype depending on the progenitor cell. Gene fusions apply to the second theory, and they occur mainly at this state, and depending on the gene fusion, I will show it later, we have like the development of a particular subtype of sarcoma. Well, I don't know if you are familiar with gene fusions, but the most famous example of a gene fusion is the chromosomal translocation between two genes. And maybe all of you know the Philadelphia chromosome in leukemia, when we have the translocation between chromosome 9 and chromosome 22, leading to a chimeric protein called BCR-ABL. which is a tyrosine kinase with a constitutive activity. And in some studies, they showed that some tyrosine kinase inhibitors can be like an effective treatment for leukemia having this gene fusion. So as you can see, we have the two genes on the two chromosomes, gene A, gene B, and we have the breakpoint, and then we have the translocation leading to an in-frame fusion gene, and this fusion gene will be later transcribed and translated into a chimeric protein. What is done at the clinic to detect these fusions is like departing from the tissue, the RNA is extracted from the tissues, and then the RNA is converted to cDNA by RT-PCR, and then by sequencing and alignment, we can identify the genes that are involved in the fusions, and we can as well detect exactly at what exons the two genes are like fused or combined. For example, you can see here that in gene A, we have the last observed exon of the gene A is the exon 4, while for the gene B, the first observed exon is the exon 6. So we can detect by sequencing the sequence in nucleotides of these exons, so we can precisely detect the fusion and the involved parent genes. So as I mentioned before, because these chimeric proteins are difficult to target because most of them are transcription factors or chromatin remodeling factors, so it's hard to develop drugs that can target specifically these proteins. So the aim is now to investigate the interactome of these chimeric protein, because if we are able to find within the interactome a protein or an enzyme that can be easily targeted, we can at least inhibit the oncogenic pathway promoting this sarcomagenesis. So I'm showing, for example, this fusion in a synovial sarcoma. It's a fusion between SS18 and SSX1, for example. This fusion gene, SS18, SSX1, is very common in synovial sarcoma. And as you can see, the SS18 gene is a chromatin remodeling factor, and it is interacting with chromatin remodeling factors, while the SSX1 is interacting mostly with transcriptional co-regulators. So when we have the fusion, as you can see, the chimeric protein is creating a truncated interacting network in which we may lose or gain some interactions, and this will aberrate the signaling pathway, promoting later the development of synovial sarcoma. In another example of Ewing sarcoma, we have this famous fusion between EWSR1 and FLE1. For example, EWSR1 is an RNA biology factor, while FLE1 is a transcription factor. So each one of them is interacting with a completely different factors or proteins. And similarly, when we have the fusion between EWSR1 and FLE1, the truncated interaction network will promote the development of an Ewing sarcoma specifically. But interestingly, when reporting or when reviewing the data about the gene fusions in sarcoma, we can see that each histological subtype is reporting a specific subset of gene fusions. For example, Ewing sarcoma is very common with these fusions: EWSR1 FLE1, EWSR1 ERG, fuse ERG, fuse ETV, and similarly for rhabdomyosarcoma, you can see PAX3 FOXO1, PAX3 NCOA1, PAX7 FOXO1 as well, which means that these chimeric proteins, because they are like common for a specific subtype, it means that they are sharing the same pathway, or they are interacting with the same partners to promote the development of this particular subtype of sarcoma. So based on this, we hypothesized that the common interactors for the chimeric proteins that are specific for each subtype are mediating the sarcomagenesis, and they can be a potential therapeutic targets. So for example, for Ewing sarcoma and synovial, we can see the specific chimeric protein for Ewing sarcoma, and they are having these common interactors to be identified. And similarly, for synovial sarcoma, we have another subset of gene fusions. They have their common interactors to mediate synovial sarcoma. So the aim is to identify and characterize what can be a common interactor between all these fusions, because if it's common between all these chimeric proteins, it means that it's important in the pathway to promote the sarcomagenesis, and hence it can be a potential therapeutic target. So the objectives of my research project is to design and synthesize of all the pertinent oncogenic fusions of sarcomas, the purification of the chimeric proteins and their interacting partners, and finally, the validation of the interactome in specific sarcoma subtype in human sarcoma tissues. So, what I mean by design and synthesis of oncogenic fusions, for example, we have like companies, and we were dealing lastly with the Thermo Fisher company. So, for example, you give them a nucleotide sequence of a gene, and they can do the synthesis of this gene for you, and they give you a plasmid that you can use for the expression of the protein in your cells. So the plan was to find what are the nucleotide sequences of these fusions in sarcoma. So I reviewed like the literature and the data found like in patients to detect or to identify these fusions, and I could find 120 fusions in sarcoma, and I defined for each fusion the 5' partner and the 3' partner. So I have a list, let's say, of 120 fusion. You can see EWSR1. It's a 5' partner that is in fusion with many 3' partners and are 4A3, ERG, FLE1, I don't know. But even that you have now the list of the genes, and you have the 5' partner and the 3' partner, you can have for each fusion between A and B different fusion types depending on the breakpoint. For example, we can see here that type 1, which is like by 50% frequency, it's occurring at the exon 8 of gene A and at the exon 4 of gene B. So, and I was finding this in the literature. For example, I have two genes, I have different fusion types. Which one is like the best to design or to do the synthesis to express the proteins? So at the end, to be a patient's representative, I decided to design the most frequent fusion type. For example, for EWSR1 FLE1, you can see that from a paper and in different patients, we have like in patient 1, we have a fusion between exon 9 and exon 4. In another patient, we have a fusion between exon 7 and exon 4, exon 7 to exon 6. So the question was, which one of these fusions or these breakpoints or these fusion types is the most frequent one to be designed? To answer this question, I occurred to databases that are specified in the curation of gene fusions. The first one is Fusion Gene Database. We have Middleman Database and COSME. And for sure, I confirmed the data of in these databases in PubMed. So at the end, for example, for EWSR1 FLE1, I can see in this table all the reported fusions types. So, and as you can see, the fusion at the exon 7 and the exon 6 is the most frequent by 54%. So at the end, I decided to design this fusion for EWSR1 FLE1. Next, as you know, for each gene, we have different messenger RNA variants. So even if I'm taking in consideration the exact exon from gene A and gene B, I can have different putative transcripts. So at the end, I designed the sequence using the messenger RNA variants that were like tightly related to gene fusions based on the databases. And I found the information as well in Cosmic and in Fusion Gene database. You can see here the accession number of the messenger RNA variant of EWSR1. So this is the messenger RNA variant that I used, for example, to find the nucleotide sequence. And later, to have a sequence of nucleotides, I combined the nucleotides of EWSR1 from its ATG, the starting codon, to the end of the exon 7, to the nucleotides of FLE1 from the beginning of the exon 6 to the end of the FLE1 to the stop codon. And a very important thing now is to confirm that this sequence, when combined, is in frame in terms of amino acids translation, because at the end, the chimeric protein will conserve the functional domains of EWSR1, and it has to conserve the functional domains of FLE1. And surprisingly, whatever you do and whatever you design these sequences, it will be always in frame. And maybe based on the literature, 99% of these fusions are in frame in terms of amino acids translation. And for sure, I confirmed as well the lengths of amino acids in the database. Later, to confirm or to validate this strategy, I occurred to sequencing data of the HMR patient that were reporting a fusion. So for example, for this patient, it was reported with a fusion between EWSR1 and FLE1. So by the sequencing data, I got this sequence that represent the junction between the two genes. So I wanted to confirm that this sequence will be at the exon 7 of EWSR1 and at the exon 6 of FLE1. So I did blast alignment of the sequence, and interestingly, I got end of exon 7 for EWSR1, and I got FLE1 at the beginning of the exon 6, which is consistent with the strategy or with the data that I found in the database or in Cosmic. So this strategy was like valid for us to design all the 120 gene fusions of sarcoma. So at the end, I could create an inventory of all the fusions. It's 122. So, and we created a library of the complete coding sequences and the plasmids of these fusions, along with their corresponding full-length genes. So it's around 250 plasmids or 250 DNA that is ready to use for expression in a human cells. And this is like an example of the data about this fusion. You can find the chromosomic modification, the related sarcoma, the function of the parent gene, what is the involved exons for each fusion, the length in amino acids, and the length in nucleotides, and as well the reference for each fusion that I designed. Now for the next step, was like to purify or to start the purification of the chimeric proteins and their interacting partners. So I had to find the best system to use to purify these chimeric proteins. So I started to investigate or to generate stable cell lines that are that can express the chimeric protein. So very briefly, this system is called the FRTTO system or the flip-in system. So for example, here I have the CDS of my sarcoma fusion. I do a recombination and then I can insert this CDS into this plasmid. Then I can transfect the cells with this plasmid uh, to uh, to generate the stable cell line, let's say. And as you can see here, when I do the insertion of my fusion inside the plasmid, let's say it's the expression plasmid, we have tags for the chimeric protein. So when the protein will be expressed, it will be tagged with flag and HA, so we can use the antibodies later for the purification, Western blot, and all of this. Uh, also, this system is an inducible system. So once I transfect the cells and I generate the stable cell line, I will have, I can induce the expression of my protein only when the cells are treated with tetracycline. So when I treat the cells, the proteins will be expressed and I will have the proteins, the chimeric proteins tagged with flag and HA, and later inside the cell I will have the interaction of my chimeric protein with other proteins, and later we can perform the purification. So after generating the cells, we expand, we amplify, and later when we start harvesting the cells, as you can see here, we are performing a tandem affinity purification mass spectrometry. So this is what's happening inside the cell. We have the protein, the chimeric protein, and here it's called the bait, which is tagged to flag and HA. And here we have the praise that are the interacting partners. And here we are detecting some kind of stable interaction or strong interaction between the praise and the bait, which means like it's not like a transient interaction between the prey and bait, but it's like the proteins or the partners are in stable complex or interaction with my chimeric proteins. So after the after harvesting the cells, we perform the immunopurification. So we perform the immunopurification using beads that are conjugated to anti-flag antibodies, as you can see, and then we elute this purification, and then we perform a double purification using the anti-HA beads, so we can to make sure that we have a good quality purification and to remove the background that we can get during the analysis of the mass spectrometry. So later, after performing this double purification, now we are capturing this bait, the chimeric protein, with the praise, and later by mass spectrometry we can identify and characterize these interacting partners. However, as I mentioned, this technique can detect only the praise that are in stable interaction with the bait, but inside the cell sometimes we have praise that they will just come interact in a transient manner with the bait, and then they will go interacting with other proteins. So it's interesting as well to identify these interactions. So to do this, we are we are using another system of purification, which is called the BioID. So in this case, we are tagging the bait with an enzyme which is called the BRA. This enzyme will biotinylate any protein that approaches the bait protein in presence of biotin. So in this case, we treat the cells with tetracycline and biotin. Tetracycline, we will induce the expression of the bait with the BRA enzyme, and in presence of biotin, this BRA will biotinylate any prey that is interacting with the bait, even in a stable manner or even in a transient manner. And later, as you know, we have this strong affinity between streptavidin and biotin. So we will use beads that are tagged to streptavidin, and then we will purify and we will purify all the biotinylated praise, even if they are not like in direct interaction with the bait. And similarly, when we do this purification and we have the biotinylated praise, we will identify these interacting partners by mass spectrometry. So for the first attempt, when we have the TAP MS, as you can see, I started first by validating the expression of my proteins inside the cells. And here I'm showing you this set of gene fusions. These fusions are found in Ewing sarcoma. This is the whole set. It's 9 fusions for Ewing sarcoma. And these are the expected molecular weights of my chimeric proteins. And it was, unfortunately, it was okay. I didn't have problems in terms of molecular weights of or the expression of my proteins. And here I'm showing you the result, or how we detect an affinity purification. So as I showed you before, we do the IP1, we do the elution, and then we do the second immunopurification, and then we detect the proteins by Western blot. And as you can see here, I have an expression of S7 in compared to GFP. GFP here, it's not tagged to HA or flag, so I will not see anything. While in S7, I'm seeing, here I am seeing the band of flag, HA, flag, and HA. And this purification will go later for the purification, for the mass spectrometry to detect the proteins that are eluted or that are captured within this purification, where I have the interaction between my S7 protein and the partners. On the other hand, I did as well the confirmation, or the verification of the expression of my proteins for the second system, the BRA system. And as you can see here, these are the expected molecular weights. It's the same subset of gene fusions for Ewing sarcoma. So in this case, I'm doing this parallel investigation while performing the tandem affinity purification mass spec, and I'm performing as well the BRA or the BioID mass spectrometry for the same chimeric proteins. So in this case, I can detect for the proteins of Ewing sarcoma the partners that are in stable interaction with these fusions, and the proteins that can be in transient interaction, so we can understand more about the signaling involved for this sarcoma subtype, for example. And here you can see the purification for the biotin labeling based. And as you can see, when we have the biotin only, we don't have the expression of the protein. When we have the tetracycline only, it will just induce the expression of the protein without any biotinylation of the interacting partners. But when we have the biotin, we will have the smears. And here, for example, when we do the streptavidin, as you can see, we have the smears. So each line of these represent a protein that was interacting with my my enzyme, or the construct of interest. Here I'm having the construct called S22. For example, it's a gene that is called FEV. So far, I didn't send the purifications for the mass spectrometry. We are in terms of purification of the other sequences, so we can send like a whole set. So I don't have like a data about any potential interacting partners. So far, because I was like adjusting the system, the expression, the design of the constructs, and in fact, it took me a lot of time to review these fusions, and especially to design the coding sequences. It took me almost 1 year to have like the library of the coding DNA, and it has been like months since like I started the expression and the purification attempts. Finally, uh, well, based on the data that we will get from the characterization of the interactomes, we will identify and characterize more the interaction networks of these chimeric proteins. We will identify the involved signaling pathways, and we can for sure generate hypotheses or conclusions on the crucial mechanisms of sarcomagenesis, so we can find some potential targets. And finally, to validate this in a human sarcoma tissues, we created a biobank, a sarcoma biobank at the hospital in collaboration with the sarcoma clinical team. So we obtained the consent from the patients. We get the tissues by biopsies or resections, and later we snap freeze the tissues, and we biobank them at -80 or liquid nitrogen. And later, for sure, we will use this data for the tissues, I mean, for RNA extraction, so we can perform transcriptomics, protein extractions. So I can as well confirm the interactions that I identified in cells. I can do the characterization as well in the human tissues, and we can use it as well for the generation of cell lines or for microscopy analysis. At the end, the most important part, I would like to thank all my supervisor for sure, all the members of my lab, and all the members of the sarcoma clinical team. In fact, it's a huge project actually, and it's a lot of work, and it's a lot of collaboration between the fundamental and the translational research. And thank you all for your attention and for your I don't know, precious time, let's say.
ميرسي كتير مصطفى على الانفورميشن والبرزنتيشن الحلوة. أكيد إذا حدا عنده أي سؤال فيكون عم يتفضل.
ما بعرف إذا كانت كتير complicated، إذا كانت كتير يعني I tried my best.
تمام، كانت كتير كانفورميشن وبرزنتيشن عن جد كانت كتير تمام.